Well, welcome to today's Safer World Design webinar. And today we're going to be focusing on the use of in silico methods, in particular QSAR approaches. And we have a very experienced expert with us to explain the ins and outs of using such methods in safety assessment, including for a regulatory purpose, with Roustem Saiakhov from MultiCASE. The presentation will be approximately 20 minutes long, and then we'll have a discussion session. You can enter your questions into the question panel, and hopefully we'll have a good discussion at the end. So with that, I hand over to you, Roustem. Please go ahead.
Thank you, Barry. Thank you for your nice introduction, and thank you to all your attendees who found some time to listen to my presentation. So the topic is use of in silico methods for assessing toxicity. And when Barry said that I'm going to cover in and outs of using QSARs, that was a little bit an overstatement. It's not much possible to do a lot of in and outs in 20 minutes. I'll try to cover some basics, give some examples. And if you will have follow-up questions, you're more than welcome to ask them after presentation, or later on I will give you contact information. So let's go. So we're going to touch base on computational aspects of QSAR models, and we're going to discuss, using some examples, how the model's output can help in reviewing process, because as probably many of you already know, it's not enough just to run QSAR. You have to look at the results, you have to make your expert conclusion, and you have to convey this conclusion to whoever you need to convey. And that really needs to be convincing. So I'm going to answer a couple of why, which, what, and how questions during this presentation. Hopefully, I will cover the most important parts. And again, MultiCASE already recorded a couple of webinars on specific topics, how to do expert review, how to address M7 problems, how to use some other models. So if you are interested in listening to those, please reach out to Barry or to us, and we will send you the proper links. First, do we need computers at all? We have this famous picture from a famous paper by Ashby and Tennant, almost from 20 years ago, and it pretty much shows that if you have those features, which are highlighted in red, that might mean that your chemical is carcinogenic and mutagenic. So if you just remember those features, maybe you don't even need to do QSAR analysis. You just identify the feature and that's it. That's at least some of them common, I would say, misconception. And here you can see the ethylene epoxy groups, you have the aromatic amines, you have N-oxides, aromatic nitrous, you have N-nitrosos, of course, and all of other things. So why we still need to use a computer? By the way, I borrowed some slides from Dr. Naomi Krulak, who is a scientific lead at CEDAR, US FDA, and with her permission, of course. And some of the slides were prepared by my colleague, Dr. Suman Chakravarti, which I was also using here. So why we are going to use a computer? Why not simply use a visual inspection? Well, if we go by the formal line Q&A document for M7, for example, says strictly you don't do any visual inspection. You need to run computer analysis. And the rationale is behind this. There are some highly complex associations which can be captured only by the models. For example, you remember I emphasized that you have epoxy group in that previous picture. So you might assume that the epoxy group, if you have it in your structure, that's it. You have a mutagenic compound, which is not true in reality. If you have long chain aside of epoxy groups, then it's not mutagenic. So from this point of view, those alerts, published alerts in this paper and some other papers, these are quite general. And the disadvantage of these alerts, they usually do not consider mitigating features or cumulative effect of multiple substituents. This long chain substituent attached to epoxy group, that would be mitigating feature, which would reduce or annihilate the mutagenic effect of epoxy group. Another example is primary aromatic amines. if you would assume that having primary aromatic group in your compound makes it 100% mutagenic, you would be wrong, at least at 50%. So it's 50-50 chance that if you have aromatic amine, that your compound will be going to be mutagenic. And what it depends on, it was a very good paper by Olberg from 2016, which actually using aromatic amine as example, showing that the whole mutagenic activity is obviously dependent on stabilization of corresponding neutronium anion. If it is stabilized, then you have increased mutagenicity. If it's destabilized, it's decreased mutagenicity. And for example, steric bulk group near nitrogen will reduce formation of DNA for this particular reason, just for simple steric effect. But in this table, you see the groups and positions which will increase mutagenic effect, and you will see group and positions which are highlighted in green, which would reduce mutagenic effect, sometimes just killing it completely. So you see how much mistakes we would make just assuming that aromatic amino group causes mutagenicity. And all these relationships were actually picked up by using the QSARs. Details are in that paper. So let's assume that we agree we have to use QSAR modeling to do correct predictions. So what is this QSAR modeling? I guess it would be a very basic statement, but just in case, I will just remind that QSAR means quantitative structure activity relationship. And the whole idea is we will try to model association between some different attributes of chemical structures, sometimes some calculated properties, sometimes just presence or absence of particular pieces of chemical structures, and their biological activities, which can be toxicity, can be something else. The whole thing is done under general assumption that similar molecules exhibit similar chemical and biological properties. Well, if you have a set of chemical of, let's say, 100 compounds, and 50 of them are positive in whatever endpoint it is, and 50 of them are negative, and there is absolutely no relationship between the structure and biological activity in this case, QSAR would not be possible. These cases are rare, but they could happen, though I did not see them in my practice. And another assumption is that the activity can be explained by chemical structure using the same aromatic amine. If you have aromatic amine group and you have a proper configuration of substituents around it, then it is mutagenic. If you have different combination of substituents, you have non-mutagenic structure. But as long as you're able to identify those factors, you can explain the activity using just chemical structures. What is an important feature of QSAR modeling? These are not simulations. The models learn from the results of actual tests, laboratory tests, or, for example, in case of human adverse effects, from the results of adverse effect databases, from everything that was reported by patients, by humans, etc. So it's all based on real results. There is no simulation values in there. So computer will examine some pieces of chemical structure and try to find those pieces which are mostly associated with particular activity, and these pieces will be called structural alerts. Of course, it sounds very easy, but in reality, there is a lot of mechanics, machinery behind that, a lot of statistical procedures, etc. I will explain it a little bit later. The same way can also identify attributes which mitigate the activity. Again, going back to aromatic amines, those fragments which will kill the activity, these can be identified by computers. And once the model is done, we can use it to make prediction of new chemicals based just on the structure. And to do that, you don't even need to have a chemical synthesize. You can test imaginary chemical. You can test a chemical which you're going to produce or not going to produce. And before you produce a chemical, you actually can test it, potential liabilities. So if you have some perfect drug candidate, as you think, and you test it for M7, and you find it to be very strong mutagen and potentially carcinogen, maybe you need to look for another candidate. So which methodologies are used to build those models? We can probably, at least I can probably split them into three groups. First is statistical-based models. So as it goes from the name, it's pure statistical. It can use linear regression, naive Bayes, partial least square, and all these methods which are listed here. And it will use a classic training set. That's a necessity. If you want to train from something, you need to have something to train from. So you have to have a set of chemicals, and strictly speaking, you just need to have only structures. and affiliated activities. Of course, the structure should be correct and the activities should be correct. That means you don't just collect chemicals, you curate them. You curate them from point of view of chemistry, you curate them from point of view of toxicity. Once you have it, you can build the model pretty quickly, and depending on which software you use to build the models can vary in interpretability of the results. Some of them can still be black boxes, some of them are very open and show everything, how the prediction was made. Rule-based models are more intuitive and more understanding. They are human expert derived. It literally goes like this. If you have aromatic amine and you have particular substructure, your chemical is going to be mutagenic. That's the rule. This rule is encoded using special ways. Sometimes it's encoded in smart code. Sometimes people are using different ways, but the rule is encoded and normally supported by mechanistic information like those bulky group close to aromatic amine, steric hindrance, that's it. It kills activity. And quite frequently it comes with citations. Of course, it's highly interpretable, but of course, also it's very time consuming to build. You don't just assemble a list of chemicals and run them through some kind of statistical routine. You actually have to find the rule, encode the rule, validate the rule, make sure that it works, make sure that you don't need to modify it, and provide all proper references. And if you have one rule, it will take 1 day to create one rule. And if you have 100 rules to create, well, you can figure out how many days it will take. Read-across approach is slightly different. It uses a classing reference set. Mind you, not training set, reference set. You just assemble a list of chemicals with their appropriate toxicity. Again, the same limitation is applicable. It needs to be reliable chemistry, and there needs to be reliable toxicity data. And you're pretty much using the similarity-based approach. You assume that if your query chemical has very closely similar analogs, you can calculate the activity of this chemical based on the activity of these analogs. Again, it might vary in interpretability. So the best case scenario, you probably can say my chemical is toxic because there are 5 analogs which are very close, and each one of them is toxic. That's why it is toxic. It will not identify any alerts. It will not give you the reason for toxicity other than so many analogs have this kind of toxicity. Those models, none of them are good if they will not follow OECD validation principles. I'm not going to go through all of them. There is a guidance which pretty much states everything about them. Briefly, it needs to be defined endpoint. It needs to be unambiguous algorithm. It can be black box. Black boxes would not follow this, would not pass this rule number 2. Ideally, it needs to be defined domain of applicability. So you should know before the test what you can, what you cannot test. The model should be validated. Measure of goodness of fit, robustness, predictivity, it should be included, should be covered. If possible, mechanistic interpretation. Sometimes it is possible, sometimes it's not. Usually the well-created model, well-validated model comes with a so-called QMRF document. It's model reporting formats, which very formalized, very well structured, and has all necessary information, very easy to read, very easy to understand if the model is good or not. So if we have very good, very well predicting QSAR model, can we use QSAR models to predict anything? Well, let's try to figure it out. What it can do normally, it can predict small and mid-sized organic molecules. It doesn't matter if these chemicals are naturally occurring or synthesized, we don't care. As long as you have a structure for this chemical and it's not huge, you can load it into QSAR model and use it as a query compound to get this or another prediction for the endpoint. It can predict particular cases, but special handling is needed. For example, if you're dealing with polysaccharide or PEGs or anything like that, or polymers, you can use shorter fragments of those. You cannot load the entire molecule. You can just use the most representative fragments. And with organometallics, you usually would require the mass modification. What we do, we just connect, let's say, in platinum complexes, we connect platinum with covalent bond with the remaining of the ligands. And in this way, we test it as a single molecule. Usually, you cannot predict huge polymers. Usually, you cannot predict inorganics because inorganics are so unique. So there is no statistical observations which can be supported by them, but they still can be part of the database supporting database reference datasets, which means there is a chance that you can predict them using read-across approach. Normally, you cannot directly predict organic chemicals which are mixtures. You have 5 or 6 or 7 components in your mixtures, you need to predict each one of them. So that's the way to predict. And assumption is, if you have 5 components, each one of them is negative in particular endpoint, the whole mixture is negative. One component of the mixture is positive, then your mixture is positive. And if your chemicals have extremely unique molecular features, which is poor structural coverage, most likely you'll get out-of-domain prediction, which from the regulatory point of view, consider as no prediction. So it needs to be resolved one way or another. Another limitation is most of the QSAR models cannot differentiate between stereochemical or geometrical, like cis-trans isomer pairs. This might be temporary limitation. In future, this situation might improve. So the final question, we talked about methodologies, we talked about what it can, what it cannot do, but what software should we use? I'm in no way can recommend you what you should not do. I can list the commercial platforms here, QSAR, DirectSAR, QSAR Applier. All of them are currently used by US FDA. I'm going to use QSAR for my examples because we are producing QSAR and I'm mostly familiar with QSAR and I obviously have access to QSAR. I don't have access to DirectSAR, etc. There are free platforms, OECD Toolbox, Tox3, EP Suite, etc. While free platforms are, what is good about them, they are free. They have to be also properly maintained. Sometimes it's not the case. And amongst available models, most suitable are and most useful would be those which are listed here. Highlighted is bacterial mutagenicity, which have a very strong regulatory support. The whole ICHM7 guidance is relying on bacterial mutagenicity models. And there is a recently released OECD guidance related to skin sensitization. So the skin endpoint is partially highlighted. All these endpoints which are listed here, or at least we at MultiCASE with QSAR support all of them and we have models for them. So what is a QSAR model? How does it look like? It's usually a combination of set of alerts and their weights and database of supporting chemicals. The example is given here. You have an equation, you have an alert, well, you have aromatic nitro, you have another form of aromatic nitro with unsubstituted meta positions, you have this chloro substitution, and then you have the dichloro. Each one of them has its own internal name, and each one of them has a weight. Those which are highlighted here in red, these are so-called activating alerts, or which presence will call increase the activity. But we also have some which are deactivating, their presence will decrease the activity, and their weights will be in negative. How do we define those weights? We use regression analysis, we use naive-based methods. We actually have a couple of publications. If you are interested, we can provide the references to those. And once you run your model, you will need to report the results. And some of the pieces of the reports are extremely important. These are highlighted in bold here. Of course, you need to do some kind of housekeeping. You need to provide a version of software, what type of the models you used, and whatever is known about this model, like cutoffs, threshold values. You cannot provide results of the prediction without actually providing predictions. And so it's the same with overall conclusion. And usually there should be confirmation that the compound which you're testing is within model domain of applicability. But what is most important, if you apply any expert knowledge or expert review to make the final conclusion, it needs to be spelled out. Usually it's done by using the analogs where appropriate. And if you, especially when you override any prediction, when model says it's positive, you say it's negative, or another way around, you absolutely have to explain it using all the output from the model plus your own conclusion. That's extremely important. There is no software which will give you automated expert review. Even if they claim to do so, it's still not correct. The expert review needs to be concluded by human expert. The software can prepare everything for this expert review, but you as a user, as a person who is responsible for the final conclusion, need to at least to have a look over. And in appendixes, usually it's raw QSAR outputs, and experimental data for all the analogs which you used. So if, for example, we concentrate on M7 endpoint and we look into the associated workflow, it's presented on this slide. Looks quite complicated, right? Except it's all implemented as an algorithm and part of our consolidator tool. So whatever you see here, it's actually implemented as a software code, and it's done for you in an automated way. And as I mentioned, the analogs are the main driving force behind all preparation for expert review. So here, for example, an example of a query compound, which is a primary aromatic amine with some substitution around. And we have an alert, we might assume it is positive, but then we start looking for alerts. And what is a specific feature of those analogs, not alerts, analogs is we don't just do visual comparison. We have a way to calculate alert environment similarity. That means situation in your molecule around your identified alert within 5 bonds distance. And this is quantitative measure. We actually can calculate similarity similar to Tanimoto similarity, but not the whole molecule, but just the area around the alert. As you can see, we have a couple of alerts which are ranging from to be very similar, like this first one. Is different only by the absence of this para-methyl group. Otherwise, everything is exactly the same. Two slightly less similar, but which is still kind of highly similar. The only analog which we do not consider here is this one, because this particular molecule has very well-known alert for mutagenicity in this case. This is highlighted in purple. And if we already have one reason for potential activity, we don't need to consider this alert. We're trying to find only so-called pure analogs, which do not have any other known alerts. So we have 4 analogs, very with a reasonable degree of similarity, alert environment similarity, and each one of them is negative. So my assumption would be this query molecule should be Ames negative. So this is the real case study. We have this query compound, 2 nitro groups, and everyone who knows a little bit about mutagenicity and toxicity would say, oh, aromatic nitric, that should be bad. And we have 2 of them that should be even worse. When we run it with our rule-based and statistical-based software, one of them says negative, another one says positive. That is conflicting results. Nobody likes conflicting results. There is one easy way to resolve them. You just go with a conservative approach. If you have at least one positive call, you say, well, it is positive. In this case, it would be wrong. When we looked into the negative, when we looked into the analogs of this chemical, we found multiple similar negative analogs. One of them is presented here as an example. As you can see, really it's very different. The only big difference, we have third butyl group here, pretty far away from those nitros, so it should not directly affect them. But nevertheless, this chemical is known Ames negative compound with fairly high alert environment similarity. Plus, we have expert role. It's all properly reported. I simply don't have a time and place here on this slide to show you what exactly the activating mitigating feature was found, but the description is in nitroaromatics with orthogroup with sterico electronic deactivation. So, as a result of expert review supported by these pieces of data which were generated by the program as a part of the report, we were able to conclude that the expert decision would be it's negative. And I verbalized my logic of reaching to this conclusion. But if it's done on paper, if it's just written as a short report, that should be enough to convince regulators that the chemical is indeed negative. If we looked into the second example, we have exactly the same situation. Rule-based system says it's negative, statistical-based says it's positive. And since we already know that in these conflicting situations, we actually need to look into the analogs, we generated the analogs, 5 of them. They are more or less within the same range of alert environment similarity between 0.6 and 0.4. Each one of them is positive. That gives us a support to the final conclusion that this particular case resolved towards positive. So it is ICH-M7 class 3. That conclusion would not be possible without generating those analogs, and these analogs are generated normally by the software, and these are part of the report. If we look into different endpoint completely, For example, acute oral toxicity, LD50. And in this particular case, the most effective way to do it is to read across. So when you run this query structure with our models, you will receive the closest, for example, 5 analogs, which are listed here. One of them happens to be exact hit, which is exactly the same compound. But for each analogs, you will have similarity degree, you will have your available information. I omitted putting toxicity values, but of course you will have toxicity values in here. And using this information, you can calculate by yourself, or program will do it for you. It will calculate weighted average out of 5 neighbors, which comes up with this value. And almost final slide here. We also used our system to evaluate human adverse effects quite effectively. It was done, well, almost 10 years ago, but it's still valid, still actual. The only difference compared to then and now, our models became even more predictive. So I guess if we'll try to do something like this today, we'll get even better results. So what we did, we assembled 870 drugs from the Saiakhov adverse effect data set and tested them with case alpha for carcinogenicity, genetic toxicity, liver, cardio, renal, and reproductive toxicity. And at the same time, we assembled 47 drugs which were withdrawn from the market and listed at the US FDA data set, and we used case ultra to find out, can we identify the reasons why these 47 drugs were withdrawn? Can we calculate those reasons, and are those calculated reasons matching with actual reasons? So the result was, I would say, encouraging. When we tested the entire CIDR set, we have quite reasonable sensitivity specificity for carcinogenicity, which are highlighted here in bold. For liver toxicity, cardiotoxicity slightly lower, but this is effect which is very hard to predict, especially when it's based on the patient's outcome. Patient feels that he has some discomfort in the heart area, he would report that he has some effect. Maybe it's because of the drug, maybe because of the weather, maybe because of the previously eaten food. It's dirty data. Renal toxicity has similar issues, and reproductive toxicity pretty well calculated results. And what is most important, I think, Case Ultra was able to correctly recognize relevant toxic effect in 43 out of 47 withdrawn drugs. Means it recognizes that these chemicals are toxic, and they are toxic for the same reason as they were withdrawn. Out of those, 9 drugs were not part of training set of any of the models, and we correctly identify them as unsafe, meaning each one of them was predicted to have one or more adverse effects for humans. So from this point of view, it should never go into circulation, and then it would never be withdrawn. Unfortunately, the producers of those pharmaceuticals were not familiar with our software probably at the time. So if they were, they probably would save themselves from this situation, from the financial loss and from all reputational loss. With this, I can conclude that properly designed and well-maintained in silico toxicology platform are powerful, reliable, and suitable for regulatory submissions, as well for exploratory activities. The keywords here are properly designed and especially well-maintained. Not enough to create a platform once. It has to be maintained because time flies, new data appears, new models can be generated, old models can be updated. It has to be maintained. And if you have questions, please ask them now, or you can address your questions to us. Here is a phone number, there is a contact information. And with this, I just have one more task left. I have to acknowledge that this presentation would not be possible without great help of MultiCASE people, Dr. Suman Chakravarti, Gianna Tiofi, Monica Giriredi, Narada Ala, and several slides from Dr. Chakravarti and Dr. Krulov's presentations were used in this presentation directly or as an inspiration. With this, I thank you for your attention, and I'm ready to answer your questions. Thank you very much.